So we'll start talking about the chromosome aberrations, our today's case studies. So this is how we display chromosomes. This is not a Photoshop. This is actually how do chromosomes look like during the cell division when you isolate them and stain them, and then we arrange the chromosomes into pairs, into the homologous pairs. How do we identify them? Pairs with the same length, chromosome with the same length. And we arrange them in descending order of length, as you can see. 1 through 22, these are the somatic pairs, and the sex pair is going to be an XX in a female or an XY in a male. As you can see, we have 2 copies of each chromosome. Look at chromosome 7, we have 2 copies. Look at chromosome 21, we have 2 copies. Look at any chromosome, we have 2 copies. Whenever you have more than 2 copies, if you have 3 copies, we call this condition trisomy, as we see in Down syndrome or Klinefelter syndrome, for example. And this trisomy happened due to a mechanism called non-disjunction. So let me come to the explanation of non-disjunction. If you remember the first part of the lecture when we discussed the meiosis, we said that the 2 homologous chromosomes have to separate from each other, and this is called disjunction. If they did not separate, this will result in one ovum containing 2 copies of the chromosome instead of containing 1 copy. And this will result, if that ovum becomes fertilized by a normal sperm, the resultant zygote is going to have 3 copies of that chromosome, and this is what we call trisomy, and this is the basis of Down syndrome, as we're going to see. So let me explain this non-disjunction in this drawing, and let's see what is going to be the results of having non-disjunction.